Greetings, all and sundry, near and far. We're now about to get into all the actual microorganisms in our treatment of an introduction to microbiology. And we'll start with a short chapter on the classification of microorganisms. And then we'll start looking in detail at all the bacteria, viruses, and eukaryotes that cause human disease, or the major ones, I should say. There's just a micrograph of Pneumocystis cerevisiae, which is a opportunistic pathogen that plagues people with compromised immune systems. At one time, I don't know if it still is considered, but at one time it was considered um, the diagnostic um, finding for people with HIV/AIDS. It's probably shown right in this context because originally it was classified based on biochemical properties as a protozoan, but it's recently been reclassified as a fungus based on its ribosomal RNA sequence, which is the standard for our current classification scheme in general. So taxonomy is the science of classifying organisms. We bumped into that a little bit early on in our introduction, and we learned a little bit about the two-name system for all species organisms. I don't know what this means. A goal of producing an all species inventory. I don't know whose goal that actually is, but as the next line indicates, um, it's sort of not only unlikely, but it's impossible to know when such a thing has been attained. But scientists for sure are interested in constantly looking for more organisms that haven't been yet identified. There's many organisms of all sorts, everything from beetles to fungi to bacteria that have not been cataloged yet on this earth. And there's people that spend their entire careers in remote areas just looking for new species to identify. Pretty cool stuff if you like that sort of thing. So there's a long history of naming schemes or organizational schemes for classifying all organisms. And I'm not going to go through all these with you, but just to show you, there's been a lot of sort of growth as we learned more and more organisms existed, then more and more kingdoms were invented in order to place them into new categories and so forth. You could see right around the time that Van Leeuwenhoek um, discovered microorganisms. A little bit later than that, right? That was the late 1600s. Then soon came some classifications that included fungi uh, and bacteria. I thought there was one before this. I guess not. Anyway, he was in the late 1600s, and then anyway, I won't go through all those. I'm tempted. I'm always tempted. We come down to today. We have, instead of 5 kingdoms, which prevailed for a long time as a scheme, we have 3 domains into which all known living organisms can be classified. And Carl Woese is the person who sort of contrived this wonderful scheme that we now adhere to for the most part as biologists. Here's the scheme in a really cool looking little drawing. Bacteria, archaea, and eukaryotes. That constitutes the 3 domains into which all organisms can be placed, and it's based on um, relatedness, ancestral relatedness. So the way this works is if you see a stem, and then something branching off, then everything following that on the branch, those are most closely related organisms. And you can see that chloroplasts and cyanobacteria are closely related because they branch off from the same stem and so forth. Excuse me. And we'll look more at some of the branches and so forth as we go along. Here's a little comparison guide for some of the properties of organisms in the 3 domains, and I don't really think we need to spend too much time thinking about this. I guess there's a few things that we already know and that we can throw in there. Bacteria have peptidoglycan cell walls. Eukaryotes have a range of different compounds in their cell walls, from cellulose to chitin. Membrane lipids, the reason that's in there is to differentiate bacteria from archaea. Archaea have different chemistry of their phospholipids that make up their membranes, but we're, again, ignoring archaea essentially entirely. Um, first amino acid in protein synthesis. When the ribosome sets up to pick its first amino acid, and in eukaryotes, it's methionine. In prokaryotes, it's formylmethionine, just has an extra organic functional group attached to it. Um, anyway, let's go on. Um, the endosymbiotic theory we've also bumped into before, and that's the notion that um, probably mitochondria and chloroplasts that we see in eukaryotic cells arose originally as free-living bacteria, which then became ensconced in larger eukaryotic cells as they developed, and now we have these cells with extra capabilities. Mitochondria are wonderful energy-producing powerhouses, but probably they were originally bacteria, and they have many properties of bacteria. They divide like bacteria. They have a circular genome like bacteria. They have a number of genes in common with some kinds of bacteria. Here's a really cool thing then on that subject. Here's an organism that's sort of like a model organism for this theory. It has a chloroplast-like structure in it, but the chloroplast-like structure actually still has a peptidoglycan wall on it, just like a bacterium. And so it's thought that this represents a sort of more recent incorporation of a bacterium into another cell. Very interesting. A little bit more on the history, probably, natural history of microorganisms real quick. Stromatolites are structures formed by communities of bacteria, and they harden into rock-like structures. And if you cut one open, it looks just like this, which is actually a fossilized stromatolite that has been dated to 2 billion years old. So bacterial presence on the Earth goes back at least to 2 billion years ago when these guys were living and producing these exact same structures. Pretty wild stuff. You're just another fossil of a filamentous prokaryote from 3.5 billion years ago. So life on Earth quite a bit older. Phylogenetics is the way of grouping organisms according to common ancestry. That's what we were just looking at. That diagram showing the three domains is a phylogenetic chart showing relatedness of organisms according to their ancestry. And this has been going on for a long time. Biologists love to classify and organize things and look carefully and examine all the species and see what seems to be most related. And initially, anatomy would be the way to look. You'd see all the rod-shaped bacteria must all be related in some way, and all the cocci must be related in some way, and all the spirochetes and so forth, and all kind of animals. And and then fossils have been used a great deal for comparing the structures that make up the anatomy of various organisms. But now ribosomal RNA is what we're using to classify microorganisms. So again, we've run into this binomial naming system before where we have a genus name and a specific epithet to define a species. So an individual organism can be defined by these two things absolutely. As we'll see, there are more different categorizations or classifications within the overall taxonomy of microorganisms. Just some examples. You can see the genus name, and then finally a specific epithet that has some particular historical importance or sign of its origin. Sometimes the genus name is honoring a particular scientist, but that's also true sometimes the epithet, such as honor this particular Trypanosoma cruzi, honoring a famous scientist named Oswaldo Cruz. So anyway, the naming scheme. Here's a little more expanded out, entirely expanded out taxonomic hierarchy. We know that there's three domains. Into those domains are kingdoms, and then phyla and so forth, all the way down to species or specific epithets. But I just want to throw out there that bacteria and archaea do not have a kingdom. So this is the scheme in more colorful and with form with examples. Archaea, no kingdom. They're just two domains. Bacteria, no kingdom. We jump right to phyla, or an individual phylum, and then there's all the different subclassifications all the way down to the individual. And an example, E. coli, Escherichia coli, we abbreviate E. coli. is the species of microorganism that we'll be talking about a whole bunch. Okay, prokaryotes. Um, they can be cultured, not always, in laboratory media. A clone, as we've already defined, is a population of identical cells or at some point identical cells that derive from a single cell of origin. So if you take and make a nice um, preparation on an agar plate and spread the bacteria out, you'll have individual cells strewn on the surface, and then each cell will give rise to a colony or a clone of like bacteria. Strains may arise within a clone. A clone may acquire a mutation right while it's growing, and then another strain may appear within that same clone. There can be many strains of a particular species of bacteria that have slight differences in properties. Zooming in a little bit on the bacteria, the domain bacteria, breaking them into some groups, and we'll be looking at we'll be ignoring a lot of bacteria. Like we won't talk too much about bacteria that have mostly environmental interest, but we'll be talking about gram-positive bacteria that fall into two different phyla: high G and C containing genomes, low G and C containing genomes, and we'll talk more about that. I'll show you in a minute. And then over here, proteobacteria represents 5 different classes that include all the gram-negative bacteria, or most. Spirochetes we see over here, which also stain gram-negative. Bacteroides that are found in large amounts in our intestines. But these green and non-sulfur and sulfur bacteria and the cyanobacteria we will probably pretty much ignore going forward. Eukaryotic species. Kingdoms of eukaryotes: animals, plants, fungi, and protozoa. And protozoa has been historically a catch-all classification. And all these can be organized or classified into clades, which is a way of showing phylogenetic relatedness, but also a clade includes essentially the percent overlap in DNA sequences. So we'll look at a cladogram in a little while. Again, now we see the eukaryotes over here, and we have Plantae, Animalia, fungi, and protozoa, protista, and that would be the kingdoms. And then we have all the rest of the classifications giving rise to, in this case, baker's yeast. Saccharomyces. Viral species. Virus are classified based on ecological niche. What are their hosts, organisms, and so forth, and how do they enter into their hosts and things like that. We'll talk a lot more about viruses in another chapter. References. This is a big topic as we're going into all the different microorganisms specifically. It's a big topic, and you need a reference for something like that. You can't put everything in your head, but we will try to put quite a few things in our heads. But Bergey's Manual of Systematic Bacteriology and of Determinative Bacteriology are important references that we'll use. I'm sure for you that are in lab, I hope you're all in lab, already know about that, because it's the ultimate source of information about and way of identifying microorganisms in the laboratory setting. So, classification is different from identification. Classification is a way of placing organisms into an organizational classification scheme, a taxonomic scheme based on, in most cases now, a ribosomal RNA sequence. But identification is different. Let's say we have a patient in the clinic who has a particular disease or a syndrome or ailment. We want to know what microorganism is afflicting them by way of infection. We need to identify it. That's a whole different process. We need to have some information we can use to recognize what is it that's in our possession at the particular moment. We're definitely not going to be in the clinic sequencing ribosomal DNA. That's just not going to be practical at all. So we have Berge's Manual of Determinative Bacteriology, and it's an enormous encyclopedia of all the different strains of bacteria that cause disease and all their particular biochemical properties. So it's a great guide if you do a bunch of laboratory tests and you gather some information about a particular bacterium, then you can go to Berge's and find out just exactly what it is. When you have your patient and you want to find out everything you can about the microorganism that's infecting their body, you as the clinician are going to send away a sample. You're not going to personally find out any of those things. You're going to fill out a form like this and send a sample to microbiology department and have them do some analysis, and you'll get back a result telling what in the world the actual species of microorganism was. So here we see a vaginal sample taken, and some of the characteristics about that bacteria were discovered, and it can be identified on that basis. Morphological characteristics are good for helping us identify. Again, we wouldn't use those for a permanent classification, but when we're just trying to identify, it's a big jump towards getting some narrowing things down. Is it cockeye? Is it bacillus? Is it corkscrew shaped, or what? What's the arrangement of individual cells? Is it strepto? Is it staphylo? Things like that. Biochemical tests are huge, and you're already, I'm sure, learning about different biochemical tests. You may still be learning about staining, I'm not sure. But if you haven't already, you'll be starting to learn about some biochemical tests you can use to differentiate between bacteria that have similar morphologies. Serology has to do with using antibodies, which are defensive proteins created by the body in response to something foreign. So antibodies can recognize specific proteins from a microorganism. So if we have a bunch of antibodies on the shelf in the laboratory that we know recognize specific proteins that are found on specific species of bacteria, they give us a great deal of power in terms of identification. And then genetics, of course. So here's a little flow chart based on a I have a little mental block about the term. It'll come to me as I'm talking. So we work our way through this through a dichotomous key. That's what I wanted to say, and we come to decision points. Take a particular microorganism and ask whether an enteric bacteria that comes from the GI tract can ferment lactose. Yes, we move to the right, and we say then can it use citric acid as a sole carbon source? If yes, move to the right again, and so forth. And if we move through this diagram, we can do only a few biochemical tests. If we find out that this organism cannot ferment lactose, we're not going to do any more work over on this side. We're not going to check their citric acid. I guess we are going to check their citric acid as a carbon source, but the point is you're going to use logic to pick the tests that are dictated by well-established strategies for identifying microorganisms. Again, maybe a better example. Fermenting sucrose is not going to be a test we're going to do over here, because as soon as we find out whether this non-lactose fermenter can use citrate as a carbon source, we can nail down, and these are all enteric bacteria that fall in the gamma proteobacteria phylum class. So that's how you do it, and you'll be identifying unknowns in the laboratory, which is truly fun. Don't let it be a chore. It's really fun to do it, and you'll be doing tests that are logical and allow you to work your way towards a final answer to identify your unknowns. Usually it's a mixture of two different bacteria. I'm not sure what they're doing this time. All right, so here's another thing that the microbiology clinical lab people can use to help answer the question. Fill out the little chart that we saw just now that the clinicians sent to them by request. It's a whole bunch of biochemical tests in Augur that can be set up all in one fell swoop. So we can inoculate. See this person holding a plastic tube in their hand with all these different culture media, different types of culture media in it, and inoculate the whole stack in one stab. It's called, with this particular microbiological inoculation tool. I want to say loop. Loop, it's not a loop, it's a, I forget. So anyway, it's just a sharp like wire. Just going to stab down through all these and let them grow. And by looking at the results from all these little blocks within this tester, it comes from Becton Dickinson, you can see down here, clinician has circled all the positive tests. And all the tests are numbered because this has been worked up into a high science. All these tests are numbered, so if you circle all the numbers and then bring down the numbers and create an index basically. From this one we see 21007 falls into this indexing scheme as Proteus mirabilis. So it's a wonderful thing that can help us identify enteric bacteria. A lot of times there's so many bacteria in the GI tract, a lot of times enteric bacteria can get out of balance and cause inflammatory diseases of the bowel. It could be that they actually get into another compartment of the body because of trauma or surgery or something. Anyway, it's a quick way to identify specific enteric bacteria. Very cool stuff. Here's some differential media, probably mannitol salt that we talked about already before, in which there's salt in the medium, which can narrow down pretty much only thing that can grow on there, or most common thing that can grow on there is Staphylococcus species. And then we have this fermentation, mannitol fermentation, distinguishing between Staphylococcus aureus, which does ferment mannitol, and Staphylococcus epidermis that does not. So there's lots of different kinds of differential media all up and down the scale right there. And that's just an example again right there. Again, serology. We can use antibodies to try to identify a microorganism. We can do it right on a slide. Mix some bacteria in a broth on a slide, add some antibodies to it and rock it back and forth. And if there's antibodies finding their antigens, recognizing something on the bacterium, it'll start to form large clumps of antibody and antigen, and you'll see this effect. This clumping effect indicates that the antibody that we added was a definite match for the bacteria, and that would help us identify the bacteria right there. Whereas here's an example of an unmatched. There's no connection between the antibodies and any antigens in the sample. And ELISA is a really cool assay used for identifying microorganisms or antibodies, or any number of things actually, but we're just narrowing it down to microorganisms. Enzyme-linked immunosorbent assay. It's called immunosorbent because we're talking about taking a plastic well in a tiny plastic dish basically, and attaching some either antibodies or antigens to the plastic, and then adding more antibodies and antigens in a stack until eventually we add one that has an enzyme attached to it that can produce a colored substrate reaction, and we can recognize the identity of a well where something was positive in there. Let me I'll show you a diagram which this will make a lot more sense. An ELISA assay there's a ton of different variations on this theme, but here's a general scheme. We can identify an antigen, meaning a protein or something from a microorganism, by putting an antibody against it that we buy from the factory. Somebody has a big farm and they make antibodies against all kind of stuff. We stick some, attach it to this plastic well. This might be a 96 or 384 well plastic plate about this big, and into this well we place antibodies and let them adhere to the plastic. Then we put in a sample there which may or may not contain an antigen. In this case it did contain a bacterium whose antigens was recognized by this antibody and stuck down. Other wells it might not. Another sample that we put in there that had a different antigen, a different bacterium, it wouldn't have stuck. And then we add a second antibody to that antigen, that foreign agent, and that we also brought with us from the laboratory. In this case, the antibody has attached to it, chemically linked onto it, enzymes, a particular enzyme, like either horseradish peroxidase or alkaline phosphatase are the two most common ones. We stick them on there, and then we add a substrate which will change color, and any wells that actually did have the antigen present will turn color, and we'll say, aha, we know the identity of that. At least we know the patient does have that particular microorganism. Another way of doing an ELISA is called an indirect ELISA. In this case, you might be wanting to say, well, does this person have antibodies against the particular antigen in their blood, indicating that their immune system has been fighting off this particular agent? So here, in this case, we're going to take an antigen that we have in our laboratory and attach it to the plastic well. And then we're going to put the patient's sample in there, and we're asking whether it has an antibody that recognizes that antigen. And so we put the patient sample in here. We see the antibody was there. It's stuck on. Then we add the second antibody. It's an antibody against an antibody. Isn't that interesting? So this is an antibody made in some animal against human antibodies. And so it sticks on, and it has the enzyme link, and we get a color change. So two cool ways. There's a bunch of different ways you can do ELISAs. You can do ELISAs with competitive assays and so forth. So here's a 96 well plate where you can see anytime you see that blue color, that means there's a positive finding. If we had a better look at that plate, we'd probably see there was some standards on the plate of different concentrations, giving a gradation of blueness. But anyway, then you put this in a plate reader, it's called, and it will quickly do a spectrophotometric analysis of the amount of blue, and it'll give us a printout of what color was found in what wells. Here's a multi-channel pipette being used to apply reagents to the 96 well plate right here. If you're in a big lab that does a ton of these things every single day, you can buy automated instruments that can do all the pipetting and washing and steps and everything in the plates automatically. Probably in most clinical labs, so it's somebody there with a multi-channel pipette where they can pick up some fluids to add to each well in a trough and then place it and pipette it into the rows that way, or columns. So here's just some familiar diseases that are diagnosed specifically by using ELISA. HIV, that's the standard test for ELISA is an ELISA test. Can also be done by Western blot. Lyme disease, bad one, emerging disease. Rocky Mountain spotted fever, been around for a while. Rotavirus, we already looked at an example last chapter of rotavirus. Syphilis and toxoplasmosis. Toxoplasma gondii is a protozoa that's common in cats. And so maybe we can test for that using an ELISA assay if someone gets infected. Many people have been exposed to it. Most people are not experiencing any disease characteristics from it. But if someone becomes immunocompromised, then they might very well be quite sick from toxoplasmosis. A Western blot, what is that? That's another really important diagnostic test for identifying microorganisms or antibodies, depending on how you set it up and run it. First, we're gonna take a sample, either from proteins from a patient's sample of some sort or another, depending on what part of the body is infected and so forth. We're going to separate all the proteins present in that sample on a polyacrylamide gel. It's another type of gel similar to what we use in the lab for growing bacteria. It's a little bit like agarose. It's just a little tougher and more fine pored gel. And I'm going to force those proteins all through the gel in an electric field, just like we do with agarose, like we talked about before. And in this case, we're separating proteins instead of DNA fragments. And so we separate all the proteins, and we do like we did with a southern blot. We transfer all the proteins onto a special kind of paper that all the proteins will stick to, nitrocellulose, or there's different types of nylons that are used. And then, so here we have paper with all the proteins on there in the exact same positions they were in the gel. And then we can probe that with antibodies that contain enzymes. They have enzymes linked to them, and we can recognize the agent of interest. So typically what's done is you take this filter, the proteins on there, and add an antibody to it called the primary antibody, and then you add a secondary antibody to the antibody, exactly as done in this case with the ELISA, only now we're just doing it on a piece of paper on a Western blot from proteins that were separated, and we find a color. In this case, we use a dye that will precipitate onto the paper or be fluorescent, precipitate onto the paper either visibly or be fluorescent. We can use a scanner to recognize the position of an antigen from some microorganism. Or if it's an antibody, we can separate. If we're looking for an antibody in someone's body's fluids, we can separate all the proteins here, and then we can use an antigen that we have brought in from the lab of a particular microorganism that we suspect of being present, and it will stick to the antibody, and then we can use a second antibody and the enzyme. So, all right, that's a Western blot. So southern blot is identifying specific DNA fragments. Western blot is identifying specific protein antigens or antibodies. Phage typing. Phage are, like all viruses, are very specific for a particular host range. Phage are viruses that infect bacteria, and each phage will only infect a certain strain of bacteria or species of bacteria. And so if we want to know what type of bacteria is infecting someone, we can plate. This plate has a white kind of sheen to it. That's the entire plate is a lawn or a solid culture of that bacteria. But we've also then infected different quadrants of this plate, of this lawn of bacteria, with specific phage. Each one of these little circles represents a dab, little sponge dab of some phage that are specific for different species of bacteria. By looking at this pattern, you can identify the bacteria of interest. Why are they clear? What happens is if the phage can infect the bacteria, it multiplies in the bacteria and then lyses the bacteria, so it clarifies an area of the plate. And so we just have killed all the bacteria here because it was positive, that phage was positively infecting the bacteria. Flow cytometry, another really great technique for identifying microorganisms. It uses a laser and it shoots cells, individual cells in a sample, in microscopic drops through the air, and shines a laser on the droplets as they pass by, and I can say, yes, this particular droplet with this single bacterial cell or eukaryotic cell had a particular fluorescent signal in it. Next one, no, no, no. Next one, yes, yes, yes. It counts up the number of cells that are positive for fluorescence or for some property of size, conduct, all kinds of properties of bacteria and of eukaryotic cells especially can be identified by flow cytometry, by this method. I'll show you a diagram. I don't know if it'll be much easier, but so here we have some fluorescently labeled cells and some non-labeled cells in a mixture, in a fluid mixture. We introduce it into the flow cytometer. It doesn't look anything like this, but it's just for conceptual purposes. And there's a special pump that can push the fluid through a nozzle so that it comes out in individual droplets with a single cell per droplet as it passes through a laser beam or multiple laser beams. And when the laser strikes the cells, it will scatter the light in a particular way depending on the size and complexity and texture and so forth of the cells, or the inherent fluorescence of the cells. Some cells just autofluoresce and are very characteristic in that way. Sometimes we use a fluorescent dye to label particular antigens with particular antibodies. So anyway, this is a machine that can just, as this, we can do maybe count 50,000 cells in maybe a minute's time or less than a minute's time as they shoot through this stream, and the lasers and the detector goes to a computer and counts up all the events, quote unquote, that occurred, and we can count cells that are fluorescent. It's a very cool process, very powerful, billion different applications for it, but here we're just talking about microorganisms. Oh, I don't know why I did that. I meant to cover up the bottom half and then it got revealed. The rest of the bottom half of this picture is talking about a flow cytometer that can, after the cells come down, they've been checked by the laser, can actually set up an electric field and divert cells with one particular fluorescent property into one test tube, and then divert the other cells that aren't. It'll shut off the electric field and let the unmarked or unlabeled or unfluorescent cells fall down to a separate tube. It can actually collect live cells, enrich live cells, but that's not usually what we're doing here in this situation. Usually we're just identifying different cells by their properties as they come through the beam. All right, genetics is a way of differentiating or classifying or identifying microorganisms, I should say. DNA-based composition. In this case, what we're going to do is take the genome of a bacterium or microorganism, hydrolyze all the bonds so that we have nothing but nucleotide bases left. We don't care that we lost the order because we're just going to count up and then say how many of those were G's and C's and how many of those were A's and T's or A's and U's. And so then we'll figure out a percent. And that's one way of distinguishing between different groups of microorganisms. What's the percent of guanine and cytosine in their genome, particularly with gram-positive bacteria? DNA fingerprinting, we already know about. RNA, ribosomal RNA sequencing, we've talked a lot about. And PCR, we'll just mention that briefly again as we go along. So DNA fingerprinting, we just take the genome of the bacteria or the whatever protozoa, digest it with an enzyme, break it into a whole bunch of particle fragments, and separate the fragments on an electrophoresis gel and agarose gel, and then look for unique band patterns. DNA hybridization we have not talked about, which is a really cool thing. If two organisms are the same, that means that their double-stranded DNA should be the same. And so if we take the two organisms and melt or separate or denature their DNA into individual strands, and then let the strands from the two opposite species, the two different test species, hybridize to each other. If they hybridize perfectly together again, that means that the two organisms were the same. They had all the same base pairs in the same order as each other. We might find in some cases that there's a partial match, partial hybridization that shows that there's related species because there's sequences that are all the same for a certain span, and then all of a sudden some areas where they're not quite the same. So that would show that there is, though, some relatedness. If there's no hybridization, obviously there's no relatedness at all. But again, great way to see if there's organisms that are very, very similar will hybridize very well with each other. And that'll give us an instant identification possibility. All right, here's a probe being used to identify bacteria. Again, here we are wondering, well, what is our patient infected with? We're gonna form a smear on a streak plate or a spread plate with a bunch of different individual colonies that arose from individual bacterial cells that came from our patient sample. Then we're going to say, well, here's some theories. We have a couple of probes because we think it's this or this, and we'll take a probe, which is just a piece of synthetic DNA with some type of a dye or a label on it of some kind. We'll probe those colonies. We'll break up the cells right on the filter, and we'll denature the DNA and then let the probes hybridize onto the DNA, and we'll see certain colonies will be positive for the particular probe. In this case, it's salmonella. We know now that there's salmonella bacteria are in this culture. Microarrays are very similar, only we're doing things on a much smaller scale. Microarrays, thousands and thousands of separate synthetic pieces of DNA can be placed on a glass or silicon wafer in a microscopic array, microscopic grid, and then we take our samples from our source, our patient sample, digest it into fragments, and then let it wash over the microarray, and wherever there's similarity in sequence, the pieces will stick on there, and they will be able to. They can be recognized by. We're first going to label them all with a dye, and then we'll see where they stuck. And we know the sequence of all the synthetic DNA oligos that are on there, and so wherever one sticks, then that gives us some recognition of what it must have been in order to have the complementary sequence. So here's a little picture of a microarray. Many individual separate synthetic sequences are present on there, and we put the patient sample over there. You might get a bunch of hits, different colors depending on the degree of homology of the unknown versus what's on the array. Very, very powerful. Produces huge data sets. There's lots and lots that goes into analyzing this kind of thing. There has to be a whole bunch of spots that you see on this microarray that are just entirely on there to help eliminate possibilities for confusion about what we're looking at. It has to be very, very quantitative and so forth. But once you get all the data back from big data analysis, you'll have some very powerful information about what the identity of the organisms was. Bioinformatics is called the whole study of big sets of data like this and statistically analyzing it and getting confidence in the kind of results you get from these kinds of studies. Fluorescence in situ hybridization is a very powerful tool to use also. We can simply take some microorganisms that are immobilized and use a fluorescent probe to find them right where they are. So here's a bunch of bacterial cells right here, and we can just bust them open and denature their DNA a little bit, just enough that the probe can get in there. And now we can see where the probe had stuck, and we know what the probe was. So we know that because it hybridized with these bacteria, we know what species they are. So it's a very powerful method. It can be used for all kinds of where you don't need to do a lot of sample prep. You don't need to probably even let anything grow. You don't have to like go in the lab and make a culture and wait for it to grow and so forth. You can just take a sample and collect the bacteria and then test with your probes by fluorescence hybridization. So again, we have the same flow chart that we looked at before, how to identify a particular enteric bacteria. What's a cladogram? A cladogram is a phylogenetic tree, meaning a diagram that shows the relatedness based on shared ancestry of different organisms, but it also shows the percent. So when we see different distances along the X sort of direction, the horizontal direction along this chart indicates the percent relatedness of organisms. So that's kind of a cool thing right there. Cladogram. It's just another way of representing taxonomy in microorganisms. So we won't spend a ton of time on that. So that's our story for chapter 10, and we will now embark finally on a very exciting series of 3 chapters in which we actually take a look at specific disease-causing microorganisms of various classifications: prokaryotes, eukaryotes, and viri.